Submolecular dysregulation due to deuterium accumulation is the cause of cancer. A thorough understanding of the causation of pathological processes is fundamental to clinical care as it informs and guides the approach to treatment. In this presentation, I will first discuss the prevailing cancer-causing paradigms upon which our current oncology standard of care is largely based. When I say paradigms, I specifically mean the two main narratives that have been the foundation of cancer treatment by most medical centers and academic institutions around the world. These have given rise to huge industrial complex involving government agencies, pharmaceutical and biomedical firms, hospitals and clinics, universities, professional societies, nonprofit foundations, and the media. The two paradigms are, the first is cells start proliferating in an uncontrolled fashion as a result of tissue damage. And the second is the genetic theory of cancer, also known as the somatic mutation theory. I will also cover other theories and conjectures of the cause of cancer. I will describe discoveries made in cancer metabolism research and touch on hallmarks of cancer. I will then discuss submolecular regulation. In fact, it is submolecular dysregulation due to deuterium accumulation that causes cancer. Submolecular regulation is the underlying mechanism foundational to the architecture of all the various prevailing theories. Submolecular dysregulation is indeed what gives rise to uncontrolled proliferation, genome transformation, the process of rapidly dividing cells into new phenotypes, what is being described as evolution, and metabolic dysfunction. Ultimately, this is a presentation about deuteronomics. The term deuteronomics was coined by Dr. Laszlo G. Boros and first introduced at the 2019 Fourth International Congress on Deuterium Depletion in Budapest. It is an interdisciplinary approach to medical biochemistry as it incorporates submolecular isotope effects known about deuterium into clinical reasoning. It is the science of how water moves in our bodies. Three decades of basic research has confirmed the importance of deuterium in living organisms. The ratio of two stable hydrogen isotopes, hydrogen and deuterium, is a key signal influencing cellular processes at the submolecular level. The transfer of hydrogen and deuterium to different structural and functional molecules is essential for maintaining normal cellular functions, nucleic acid structures, and protein integrity. Deuterium availability regulates cell growth. This 1993 paper by Shumyae et al. Investigated the role of deuterium on living systems. They did cell line and mouse experiments and demonstrated that deuterium in fact controls cell growth rate and is crucial to the start of cell proliferation by modulating DNA structure and size. This is data that actually holds up in human trials and translates to human health as well. To provide context for the significance of this isotope ratio, the concentration of deuterium in our body is 2 to 4 times higher than the normal amount of circulating blood glucose. Deuterium is present 5 times higher than calcium and 10 to 18 times that of magnesium. When the concentration increases to levels above a relatively stable equilibrium, exceeding our physiological and biochemical capacity for deuterium, submolecular dysregulation is the result. Depletion can reverse dysregulation, as has been demonstrated in the research. Downstream effects of submolecular dysregulation are many and they vary greatly. It is those effects that have been pulling research scientists in all directions. Many contradict one another. Integral to submolecular regulation is the ability to efficiently fractionate, or what we call discriminate, deuterium. Excess deuterium in mitochondria is what drives submolecular dysregulation, leading to cancer. This knowledge has largely been ignored. This new science is now being taught in a graduate course at the Vrije Universiteit in Amsterdam since 2022. We just completed the 2023 January course. It was fantastic. Okay, so disclosures and my disclaimer. I am a naturopathic doctor who has been looking for answers, not as a research scientist or an oncologist. I got to medicine actually later in life after a few other careers, not having been shaped by the entrenched academic institutions at an early age is perhaps why I question so deeply without reservations. Trying to understand why we do what we do in medicine started with the birth of my daughter. I have not stopped questioning since, and that is what got me here to this point. I want to start by declaring my deep respect for all the researchers, doctors, clinicians, scientists that have come before me. I admit that I am often humbled by what I learn, realizing how much there is to know and that we only find out what we did not know when we learn new information. Staying open, realizing I misunderstood, I lacked crucial details, giving space to new information, and the ability to recalibrate or rethink are qualities essential to progress. I try as hard as I can to do exactly that. None of this is medical advice. Please discuss any and all concerns with your doctor. Paradigm 1. Cancer is a disease of excessive growth. Cancer begins when cells acquire the ability to grow uncontrollably and ultimately invade and damage the body's normal tissues. Cancer development happens in multiple stages, from precancerous changes to malignant tumors. This can be a result of an injury or a toxin like smoking or radiation, chemicals, certain types of light, viruses, etc. The treatment approach is to remove and/or kill the abnormal cells with surgery, chemotherapy, and radiotherapy. Paradigm 2. In 1914, Theodore Boveri published On the Origin of Malignant Tumors, widely acknowledged to be the seminal publication in cancer pathogenesis. Boveri proposed two theories. The first, proliferation is the default state of cells, and the second, cancer is a cell-based disease. He concluded that cancer is a problem of cell proliferation and that cancers are due to an abnormal chromosomal rearrangement that eliminates a portion of chromosomal material whose function it is to inhibit cell proliferation. This was a theory. The actual structure of DNA had not been discovered yet. Shortly after Boveri's death in 1915, R.C. Whitman introduced a notion that the cancer cell was a mutated cell, and coined the idea of a somatic mutation to explain what Boveri had implied in 1914. This is when the somatic mutation theory was born. The premise that cancer is a cell-based disease in which acquired DNA mutations affect the control of cell proliferation remains the dominating dogma today. It continues to be put forth as an authoritative while lacking substantial grounds. As it has been deemed as an unquestionable fact, it has determined the direction of cancer research and treatment approach for the last 50 years. There are indeed familial or inherited cancers, but they are relatively rare, accounting for approximately 5 to maybe 10% of all cancers. And given the right conditions, maintaining mitochondrial deuterium depleting function, these cancers will likely not develop. With the sequencing of the human genome, we expected to get all the answers. In fact, we got very few. In 2022, the final 8% was completed. The project cost $2.7 billion and was funded primarily by the US Department of Energy, the NIH, and the UK's Medical Research Council and Wellcome Trust. It turns out just a few forms of cancer can be attributed to a single mutation. For example, 80% of chronic myeloid leukemia is caused by a single chromosomal translocation of genetic material between chromosomes 9 and 22 that is thought to occur spontaneously. The number of mutations found in any cancer can vary from a handful to many more, usually hundreds or thousands. In metastasis, the same patient and in different parts of the tumor, we will find completely different mutations. Memorial Sloan Kettering conducted analysis of 25,000 patients over a period of 6 years and explored the question of whether it's possible to look at an individual tumor and, based on its genomic profile alone, predict its precise future metastatic trajectory. The answer is clearly no. The study was revealing in other ways. The data showed that metastatic disease is genomically entirely different from the primary disease. They analyzed over 50 tumor types and close to 100,000 metastases. A 2019 PLOS paper takes a different approach to finding out why someone develops cancer. They designed what they call the multi-combination or multi-hit model of carcinogenesis. They conclude that there is no single combination of mutations responsible for all instances of cancer, even within a cancer subtype, and that carcinogenesis is a result of one of many possible combinations of somatic mutations. So instead, they count the number of mutations that give rise to a certain cancer. Another important clue of the failure of the somatic mutation theory is the fact that cancer patients have lots of mutations, but so do cancer-free people. A 2012 detailed analysis of 31,000 plus cancer cases concluded that the vast majority of, if not all, aberrations that were observed in the cancer-affected cohort were also seen in the cancer-free subjects. A clear example of the debatable relevance of mutation profiles in cancer comes from this systematic review of the Institute of Inflammation and Aging out of the University of Birmingham. They demonstrate that of the 17,371 human genes appearing in at least one paper in PubMed, 87.7% also appeared in at least one paper mentioning cancer. This impressive result tells us that practically any gene is associated with cancer, which is most probably the case. Looking at single gene explanation is devoid of any heuristic power. The reality is that there are millions of mutations. The Sanger Institute's COSMIC group is responsible for the catalog of somatic mutations in cancer. They released their most recent version last summer, 2022. The Pan-Cancer Analysis of Whole Genomes Consortium of the International Cancer Genome Consortium, they characterized mutational signatures using 84,729,690 somatic mutations from 4,645 whole genomes. That encompasses most types of cancer. 84 plus million mutations. Then there are those who, after a cancer diagnosis, undergo extensive genetic analysis and we find no genetic mutations at all. This genome-wide association studies of cancer shows that 66 to 80% of breast, colon, and prostate cancer cannot be explained by genetics. How we can explain these findings is that the 84 plus million different mutations, epigenetics, and non-mutational cancers are due to the genome transformation as a result of deuterium accumulation. This is why we call deuterium an oncoisotope. DNA synthesis takes place in all cells through the process of DNA replication and is changed by deuteration. The kinetic isotope properties of deuterium will affect the DNA and the RNA shape, function, and size. Nucleotide and histone modifications can be altered by deuterated methyl groups. The increased bonding strength affects the efficiency of DNA methyltransferases and DNA repair enzymes. If insertion of a deuteron into the DNA backbone happens more frequently, the cell will end up with additional DNA in the cytoplasm or nucleus or both. This is called aneuploidy. A normal human cell wraps all newly synthesized DNA in 46 chromosomes before dividing. If there's too much DNA, the cell will produce additional chromosomes to wrap it all up neatly. Again, this is aneuploidy or cancer. For example, a pancreatic cancer cell, like a MIA-PACA, will have 52 chromosomes. A metastatic lung cancer cell, number 60, may have 61 chromosomes. There is a line called H441. This is due to excess deuterium present in the cell. In fact, 90% of tumors have gained or lost chromosomes. Copy number changes far exceed the proportion affected by sequence changes. Deuterium can increase copy number, structural chromosomal abnormalities, and contribute to genome instability. Aneuploidy arises through chromosomal instability, what we call SIN, by the persistent loss and gain of whole chromosomes. Chromosomal rearrangements occur through chromosome structure instability as a consequence of improper repair or DNA damage. These images from a 2013 paper called Chromosomes and Cancer Cells show this with color. Compare the chromosome number and arrangement between the two glioblastoma cells from the same cell culture. DNA strand breaking by hydroxy radicals depends on the accessibility of specific bonds on the DNA backbone. In 1998, original data from a team of the Department of Chemistry, among others, at Boston University published the effects of deuteration of specific carbons on DNA. The fifth and fourth carbon are most sensitive to deuteration, and the table shows the increase in strength required to break such a bond. In 2014, they showed a similar data on deuteration of RNA. The colorful graph shows the kinetic isotope effect that deuterium has on the reaction. It becomes 2.6 to 2.1 times harder to break these bonds. So this changes the reactions entirely. The treatment approach for this second paradigm has been to develop targeted gene therapies, immunotherapies such as monoclonal antibodies and CAR T therapies, signaling pathway inhibitors such as CDK4/6 inhibitors, BCR-ABL, tyrosine kinase inhibitors, PD-1, PD-L1, CRISPR, Yamanaka factors, and the already failed Sleeping Beauty transposon system. The Journal of American Medical Association published in 2020 a comparative effectiveness study of 92 novel cancer therapies approved for 100 indications over 17 years. They concluded that the therapies were associated with substantial tumor responses, but prolonged median overall survival by only 2.4 months. Unfortunately, this increase in median overall survival time is negligible, especially compared with submolecular regulation with deuterium depletion. We hand these drugs out like water, but have no idea how they function in the body. We only know how they behave in rodents. It's like throwing spaghetti on the wall and see what sticks. This is a quote from Thomas Marin, the assistant director of the early phase and immunotrial therapies at the Tisch Cancer Institute, in a presentation I found on YouTube. This is an actual slide he used to illustrate his point. I did not add that plate of spaghetti. He did. Some research scientists already knew this in 2005, and I quote, "Although new drugs are continually designed and tried, it seems inevitable that genetic and signal protein targets pose too broad flexibility and variability, often changing target characteristics and thus escape treatments, turning magic bullets into rather wandering bullets." The reality is that many of these drugs have serious, sometimes fatal side effects. How does this continue to go on, you may ask? Regulatory capture of our three-letter government agencies plays a direct role in the ongoing marketing efforts. A drug's review and approval process are often expedited while based on incomplete data sets, as pharmaceutical industry-funded patient advocacy organizations demand and receive faster drug approval and easier access on incomplete data sets. A 2021 British Medical Journal article identified many drugs that received accelerated approval and then failed to improve the primary endpoint in post-approval trials. They still remained on the market for years afterwards. Another factor is that the randomized clinical trials in oncology have evolved since its widespread adoption in the 1970s. Contemporary oncology randomized clinical trials have significantly changed and now largely measure by assumed surrogate endpoints and are almost exclusively funded by the pharmaceutical industry. Assumed surrogate endpoint means that the trial no longer reports on the overall survival time or quality of life improvement of the patients, which is what patients ultimately care about, real results. It's not just cancer drugs. In fact, most healthcare interventions are not effective according to high-quality evidence. The Journal of Clinical Epidemiology published this original article on April 18, 2022. Of 1,567 eligible interventions, only 87, just 5.6%, had high-quality evidence supporting their benefits. There was statistically significant evidence for harm in 8% of these. This means more evidence that medical interventions cause harm than benefit. This study included cancer drugs. Somatic mutations are not the cause. I quote, "The clinical reality is that it is not single genes, but rather the properties of aneuploid-based methylated networks that allow metastatic cancer cells to explore novel niches in different genetic backgrounds and to rapidly become resistant to drug-based therapies," states George Gabor Miklos in Another Misstep in the War on Cancer. He is correct. This is due to the accumulation of deuterium affecting those metal groups. There have been numerous papers published over the last several decades that have attempted to refute, dispute, and take down the somatic mutation theory, as clinical data show little support for the theory measured against any patient outcome data. Before I get to submolecular dysregulation caused by deuterium accumulation, I just want to discuss a few other theories. This bioinformational crisis in cancer research led to the blossoming of the evolutionary theory of cancer. What Dr. Jason Fung calls Paradigm 3.0 in his latest book, The Cancer Code, builds on both the evolutionary theory and the atavistic theory. He writes, cancer is improbably a backward of evolution or atavism toward the single-cell organism from our evolutionary past. His book is a paradox to contend with. Fung does an excellent job describing how the genetic paradigm has failed and then makes a case for how these same mutations by evolutionary force through Darwinian selection and selection pressure cause cancer. Atavistic is derived from atavus, meaning ancestor. The atavistic model posits that cancer cells have reverted to doing things that our cells used to do a long time ago, namely survive in the absence of oxygen. It postulates that cancer initiation and progression represent reversions to ancestral unicellular phenotypes. The missing piece in their theory is that when mitochondria fail within a cell due to deuterium, the cell loses function. It may seem that this is reverting back to unicellular organism, but that's not the case. He was not the first. Peter Noel, Mel Greaves, seen as the father of the evolutionary theory of cancer, and many others have published papers and books since 1976. Their premise is that cancer is the result of clonal or somatic evolution, which is explained as the process of sequential acquisitions of vertically transmittable genetic epigenetic elements in multicellular organisms. Evolutionary forces are the driver. Selection pressure determines the heterogeneity of the tumor, meaning the diversity of the mutations. It's hard to imagine that a self-terminal disease process has anything to do with the survival of the fittest. This whole new focus of cancer as an evolutionary process comes just as Darwinian theory of evolution is shaking in its boots, as new research is clearly invalidating his poetic take on how organisms evolve. The elaborate, colorful evolutionary theory of cancer offers no solutions, just more mutations and confusion. There are more theories. The trophoblast theory, the tissue organization field theory, the brand new egg cell genetic program theory, the stem cell origin of cancer theory. There are stem cells, and there are stem cells that have accumulated deuterium, driving them to divide them. What unifies all the previously discussed theories is that deuteronomics, the science of how water moves in our body, was not ever considered. Our current treatment paradigm is obsolete. I quote, "A new paradigm should reflect that cancer is a systemic disease rather than an invading organism or mere clusters of mutated cells that need to be eradicated." It is time for a common sense revolution in oncology where we can prioritize treatments that meaningfully improve survival and quality of life. Cancer metabolism. Metabolism is integral to submolecular regulation. That cancer is a submolecular process associated with electron transport abnormalities and protein dysfunction was first introduced by Albert St. Georgi in The Living State and Cancer in 1979. The understanding that cancer is a metabolic disease started when Otto Heinrich Warburg, 100 years ago, observed and published what is known as the Warburg effect. Recent advances in metabolism allowed us to follow the precise metabolic flux or movement of precursors and substrates. Using tracer-based metabolomics, important mechanisms and pathways have been identified. Metabolic profiling involves labeling common precursors, such as glucose, lactate, or acetate, with an isotope so that the pathway this molecule travels can be traced. They use mass spectrometry and stable isotopes, such as carbon-13 or deuterium, to observe and trace metabolic pathways for gluconeogenesis, glycogen synthesis, lipid synthesis, the pentose and TCA cycle metabolism, among others. As a result, non-oxidative pentose phosphate pathways and their direct role in ribose synthesis in tumors was established as the cause of cancer based on the model of cellular glucose metabolism in 1998 by Boros et al. This was the first of many excellent contributions to the field of metabolomics coming from Dr. Laszlo G. Boros and his mentor, Dr. Y. Nang Paul Lee at the UCLA School of Medicine in Torrance, California. 2 weeks before his untimely passing, he made this profound statement: "The ultimate constraint of cell metabolism is deuterium." In 2002, metabolic profiling elucidated the mechanism of the anti-proliferative action of imatinib, also known as Gleevec. Rather than an unknown signaling pathway inhibition mechanism that was claimed, imatinib controls flux of metabolites, reversing the metabolic transformation observed in the chronic myeloid leukemia cells. Imatinib limits glucose metabolizing enzymes by improving the mitochondrial functions via enhanced fatty acid oxidation and metabolic water production. In his 2005 metabolomics paper, Boros wrote, "As metabolic profiling allows new targets to be discovered, their promise is that these targets will have significantly far less flexibility and variability to escape treatment than do genetic and signal protein targets." This statement was prescient, as gene mutations and pathways diversions indeed cause widespread drug resistance. Genes always mutate. Pathways can divert. A well-known series of papers that have attempted to create an organizing principle for rationalizing the complexities of neoplastic disease, in the author's own words, are the hallmarks of cancer review papers. There are now 3 in the series, and they have over 100,000 citations altogether. The first paper was published in 2002. The second update, entitled Hallmarks of Cancer: New Dimensions, was published in 2022. The 2000 paper introduces their 6 core hallmarks. Both the 2011 and the 2022 have added 4 emerging and enabling characteristics each. By definition, a hallmark is a defining feature, a distinguishing characteristic that is part of every phenotype-related cellular behavior, but none other. A single feature, not 14. Additionally, the hallmark features contradict one another. In 2022, Hahnemann stated that genome or DNA instability and mutation is a fundamental component of cancer formation and pathogenesis. Then introduces an emerging hallmark called non-mutational epigenetic programming, describing mutation-less cancer. The 2011 paper added as an emerging hallmark deregulating cellular energetics, also referred to as reprogramming energy metabolism. I quote, "Altered energy metabolism is proving to be as widespread in cancer cells as many of the other cancer-associated traits that have been accepted as hallmarks of cancer." This realization raises the question of whether deregulating cellular energy metabolism is therefore a core hallmark of cancer cells that is as fundamental as the 6 well-established core hallmarks. In fact, deregulating cellular energetics is the hallmark of cancer. They confirm that with the 2022 New Dimensions update. This is submolecular dysregulation. I am not alone in making these statements. Many others have. In a 2022 paper, Cancer as a Metabolic Disorder, the authors state that according to the metabolic theory of cancer and somatic mutations and all other hallmarks of cancer are downstream epiphenomenon occurring from the initial disturbances of cellular energy metabolism. Furthermore, in their 2011 Hallmarks of Cancer, The Next Generation, the authors stated on page 15, a functional rationale for the glycolytic switch in cancer cells has been elusive. Actually, we did know this. That same year in 2011, original data published in Cancer Cell clearly implicated a deficiency of the mitochondrial metabolic enzyme fumarate hydratase as the cause of the glycolytic shift. They also demonstrated how restoration of the enzyme activity reversed glycolytic and tumorigenic features of the fumarate hydratase deficient cells. A 2014 PLOS publication further details how fumarate hydratase deficient kidney cancer cells undergo metabolic remodeling with changes in mitochondrial respiration, glucose, and glutamine metabolism. These changes represent multiple biochemical adaptations in glucose and fatty acid metabolism that supports malignant proliferation. The authors again demonstrated that the restoration of the fumarate hydratase function leads towards a normal metabolic phenotype rather than a lactic fermentation-driven Warburg-like phenotype. Excuse me. What this data tells us is that by restoring metabolic water recycling pathways, we can reprogram and reverse the cancer's phenotype of transformed cells. In 2016, another landmark paper was published that explained in detail how submolecular dysregulation leads to cancer, entitled Submolecular Regulation of Cell Transformation by Deuterium-Depleting Water Exchange Reactions in the Tricarboxylic Acid Substrate Cycle. This is the paper that identified the precise mechanisms by which our bodies keep deuterium away from the mitochondrial matrix. Years earlier, Olgan described how deuterium damages the delicate ATP synthase nanomotors of mitochondria. The authors state the role of deuterium in biology is a potential missing piece in the elusive cancer puzzle, explaining cancer epidemics in Western populations as it seemingly correlates with excessive deuterium loading from processed carbohydrate intake in place of natural fats consumption. These findings and the application of tracer-based metabolomics have provided new insights into the Warburg effect. In the cancer process, glycolysis can no longer deplete deuterium because there is no deuterium-depleted water available in the mitochondria and/or cytoplasm. Deuterium destroys proton tunneling. Hydrogens stop moving via tunneling, and they don't deliver energy through the nanomotor movements. Protons cannot get into the matrix, and hydrogens pile up outside the mitochondria that still are residing on lactate. This is the Warburg effect, the single hallmark of cancer. These are not theories, these are facts. Mitochondrial health determines cellular health, not genetics. Thomas Seyfried, in his 2012 book, Cancer as a Metabolic Disease, details nuclear cytoplasmic transfer experiments that started as early as 1982. Essentially, what all experiments did was they placed a nucleus of a cancer cell in the cytoplasm of a healthy cell to see if it would become cancerous. It did not. They also placed a nucleus of a healthy cell in a cancer cell to see if it would restore the cell. It did not. The nucleus contains the genetic material, which contains all the alleged mutations. These experiments demonstrated conclusively that the somatic mutation theory is incorrect. Let's briefly cover a few definitions of key terms. The TCA cycle is the chemical conversion of carbohydrates, fats, and proteins into metabolic water and carbon dioxide, which also generates energy in the form of ATP and heat. The TCA cycle leads to either the net production of intermediates, which we call anaplerosis, or their net consumption by cataplerosis based on the mode of its operation. Glycolysis is the pathway employed by all the tissues for the breakdown of glucose to provide energy and intermediates for other metabolic pathways, including the TCA cycle. A phenotype is the observable characteristics or traits of an organism, for instance, the healthy cell or a cancerous cell. Phenotype and adaptation depend on mitochondrial metabolism. It is the health of mitochondria that determines the health of the cell and its fate, and this depends on its ability to produce deuterium-depleted metabolic water and the efficiency of the TCA cycle to keep deuterium out of the mitochondrial matrix. This primarily happens in glycolysis via isomerization reactions and the TCA cycle through water exchange reactions, providing intact mitochondria. If the deuterium depleting functions of mitochondria are defective or overburdened by any number or combination of factors, submolecular dysregulation and cancer are the result. The figure illustrates beautifully how, due to deuterium accumulation, mitochondria become enlarged, misshapen, and lose their cristae. This is when submolecular dysregulation sets in, causing cancerous transformation, regardless of the status of the nucleus. After restoring deuterium-depleting water-producing and recycling functions of the cancer cell, it differentiates into a healthy cell. The fumarate hydratase data informed us that if we restore the metabolic water recycling function of the enzyme, we restore dysfunctional mitochondria and reverse the cancerous transformation process. What is the function of fumarate hydratase, you may ask? There are 8 enzymatic reactions in the TCA cycle. 3 of these reactions add deuterium-free water to TCA cycle intermediates from water that is produced in the mitochondrial matrix, also known as metabolic water. Citrate synthase is the first of the 3 enzymes involved in these water reactions, as it adds 1 water molecule to form citrate from oxaloacetate and acetyl-CoA. The second enzyme is aconitase hydratase or aconitase. This is a 2-step reversible reaction and involves dehydration followed by hydration. This is a water exchange reaction. 1 water molecule is removed and replaced with a deuterium-depleted water molecule from the matrix. The third enzyme involved in adding water is fumarate hydratase, which catalyzes the reversible hydration of fumarate to malate. If fumarate hydratase is deficient, submolecular dysregulation sets in, causing cancerous transformation. To understand the electron microscope images and figures I will display next, let's cover a few more terms. HLRCC stands for hereditary leiomyomatosis renal cell carcinoma. UOK262 is the name of an immortalized cell line. UK262 cells are derived from a patient who had aggressive hereditary leiomyomatosis associated recurring kidney cancer. UK262 wild type, WT, is the cell line restored to a wild type for normal fumarate hydratase depleting functions, which means the normal healthy phenotype of the cell. 117C4 and 7860WT are non-cancerous renal cells or they're healthy kidney cells. 3-NPA is an inhibitor of the TCA cycle enzyme succinate dehydrogenase. Now, these are images from the 2011 glycolytic shift paper that I mentioned earlier. The arrows point to the dysfunctional mitochondria. You can see swollen mitochondria with severe fragmentation to the cristae in the cancer cells. On the two left photos, similar to the abnormalities found in the non-cancerous renal cells that were treated with the succinate dehydrogenase inhibitor. This is another mitochondrial enzyme as well as complex 2 of the electron transport chain. The middle images are of healthy, untreated cells and show smaller mitochondria where the cristae are visible. These are functional mitochondria. These experiments demonstrate submolecular dysregulation. Healthy cells that lose depleting function transform into cancer cells. This next graph from the same paper took it one step further. To examine tumorigenicity of the cancer cells, the UK262 cells were injected subcutaneously into nude mice. Palpable tumors developed within 32 days with a tumor doubling time of 17 days. Animals injected with the UK262 wild type did not develop tumors, indicating that fumarate hydratase dysfunction was required for tumor growth. This graph shows a typical organic acid test I order for patients. The gray bars are the normal healthy kidney cells and the red, the cancer cell line. Notice how in fumarate hydratase deficient cancer cells, fumarate piles up while no malate is produced. On the right, the data shows that in the gray normalized cancer cell, the wild type in which fumarate hydratase function is restored, fumarate is readily catalyzed into malate. This is a real-life organic acid test result from a patient with a succinate dehydrogenase deficiency due to homozygous variation of 2 of the 4 succinate dehydrogenase genes. This allosterically affects the other enzymes, leading to submolecular dysregulation and tumor formation. This causes the same dysfunctional mitochondria as I showed on the previous slide, the normal cells that were treated with 3-NPA, a succinate dehydrogenase inhibitor. Genetic mutations only matter if they compromise deuterium depleting functions. The experiments I discussed can be described as the fumarate hydratase mutation model. Thankfully, these mutations are rare. What is not rare and the cause of most mitochondrial dysfunction is the lack of oxygen due to old age, COPD, damage to or glycosylation of circulating red blood cells, underlying metabolic diseases like diabetes, obesity, and deuterium overloaded mitochondria in all kinds of cells in any tissue, and many other factors which I will discuss next. Natural food plus oxygen gives rise or equals metabolic depleted water and CO2 and ATP or heat and heat. So this is what it's all about. The lack of oxygen directly affects the amount of deuterium depleted metabolic water produced in mitochondria. The 2014 PLOS metabolic programming study also determined the differences in the oxygenation rate and extracellular acidification between the UOK262 cancer cells and the UOK262 wild-type cells. The wild-type cells had a 4-fold higher oxygenation rate and 2-fold lower acidification rate. This is significant. Oxygen consumption directly relates to the amount of deuterium-depleted water produced in mitochondria, and the extracellular acidification relates to the water piling up, known as hydropic degeneration. This is metabolic crowding. TCA cycle metabolites back up and pyruvic acid goes down the lactic acid pathway. Again, this is the Warburg effect. In the fumarate hydratose model, which is rare, they've demonstrated that after we restore the metabolic water recycling function of the enzyme, we restore dysfunctional mitochondria and reverse the cancerous transformation process. In clinical care, these genetic therapies cannot accomplish this. It's not possible to repair genes in all somatic cells, as we cannot switch genes around in every cell. What can compensate for dysfunctional mitochondrial enzymes and restore mitochondrial enzymes and mimic mitochondrial function is to deplete deuterium in all the ways we currently know how. Restoring submolecular dysregulation. We approach deuterium depletion from all possible pathways. Let's start with the fuel we provide our bodies. First and foremost, it's the quality and kind of food you consume. Focus on those that are low in deuterium. This means a seasonal and local, low-carbohydrate, well-raised, meat-based or ketogenic type diet that matches your heritage and preferences. Choose the best available sources for all the food you consume. Fats should be as unprocessed as possible. Proteins ideally come from animals that were grown slowly and respectfully in their preferred habitat and fed the best possible diet. Vegetables should be pesticide and herbicide free. Avoid industrial fats such as soy, canola, corn, cottonseed, safflower, sunflower, grapeseed. Instead, include grass-fed butter, lard, tallow, suet, duck fat, coconut, and olive oil. The importance of the quality of the food cannot be overstated. Grass-fed meats and dairy are much lower in deuterium content compared to grain-fed animals-derived meats and dairy, as shown in this 2022 metabolomics paper. Next, I want to briefly cover what contributes to deuterium accumulation. This flowchart shows some of the key factors that play a role in the accumulation of deuterium levels in our bodies and the effects that they have. Water quality and quantity, fat quality and quantity, and light spectra are the primary inputs that determine deuterium levels. Omnipresent glyphosate, electromagnetic frequencies, and inorganic metals affect submolecular regulation as well. Lack of sulfur and essential trace minerals further impair function and reduce metabolic water synthesis. An imbalance in all of these causes changes in the microbiome, allows for viral and microbial hosting. Deuterium is a growth factor for these organisms, and it causes inflammation, alters vitamin D and A metabolism, causes genome transformation, and metabolic crowding. Submolecular dysregulation due to the accumulation of deuterium is at the heart of cellular transformation causing cancer. The approach to treatment and prevention of recurrence of cancer is to address all those factors we know contribute to submolecular dysregulation. These include, and by no means are limited to, the standard industrial diet based on corporate agriculture, the inferior quality oils, in particular the ones that I already mentioned, inferior quality of animal and vegetable proteins, and meats that contain synthetic growth hormones, GMOs, antibiotics, dyes, chemicals. Lack of spring water that is low in deuterium, but also excessive hydration with fluids that are high in deuterium. The whole premise that we need to be consuming half our body weight in ounces in water every day is not based on any biochemical or physiological facts. So I am actually an advocate of not drinking that too much water. Impaired mitochondrial function due to genetic variation in the genes for the mitochondrial enzymes. And these are tests that can be done, and we can look at that in detail. Insulin resistance and metabolic syndrome. Dysregulated iron metabolism is another big factor. Dysregulated calcium metabolism, which of course we are all affected by the massive amounts of electromagnetic radiation that alters this calcium metabolism in particular. The lack of important minerals, iodine, magnesium, copper, all of them. The microbiome imbalance presenting as a lack of diversity and an increase in pathogenic microbes. So deuterium feeds the pathogenic microbes. It's what they proliferate on. Lack of sunlight and spending time in nature and working night shifts and being in artificial light at night changes very much how our bodies respond. Thyroid dysfunction, stress, lack of exercise or movement, and of course increasing age. And as I already mentioned, the excessive ionizing and non-ionizing radiation, and this includes ultrasound technology. Electromagnetic radiation, glyphosate replacing glycine and inhibiting many biochemical processes, including completely disturbing deuterium depleting functions of the cells. Environmental toxins, plastics, heavy metals, excess alcohol intake, dental procedures such as root canals and amalgams, biologics and mRNA and DNA-based therapeutics, history of oral contraceptives or hormone replacements, history of steroid use, and a lack of natural infections in childhood. Approaching management and treatment of cancer based on addressing submolecular dysregulation by targeting metabolic pathways in every way that we know today offers clear strategies to the management and prevention of cancer. Deuteronomics is revealing new mechanisms by which we can improve our cellular energy metabolism, our protein integrity, in order to reverse submolecular dysregulation every day. To facilitate cellular reprogramming and reverse submolecular dysregulation, which will increase metabolic water production and bring the body in coherence, we have created a success list of aspects of daily life to focus on and integrate one by one where needed and possible. So consider your water, the deuterium content, the quality, the quantity. Consider fasting, both intermittent and with clear liquids. Consider the light of your light environment of all the spectra and intensities, including photodynamic therapy, photon biomodulation, sunlight, UV light, and infrared light. Adhere to circadian rhythms. Include movement, acupuncture, massage. Consider temperature flux, including soaking in enhanced cold water baths and sauna, electromagnetic frequencies mitigation. Optimize your sleep environment. Enhance your oxygen environment with algae and magnets, and try different breathing technique. Realize that oxygen is one of the 2 rate-limiting factors of metabolic water production besides protons. Add deuterium scavenging botanicals if needed. Lastly, there's no question that deuterium-depleted water can reverse submolecular dysregulation very rapidly and efficiently. It can be used as wellness water or as a therapy, meaning the ppm you consume is based on the optimal physiological levels and determined based on weight, diagnosis, and consumption. For those who have been diagnosed with the cancerous process, adding deuterium-depleted water to other therapies has shown to increase the overall survival time 3 to 5-fold. So the deuterium-depleted published mechanisms of action include all of the following. I'm not going to read them off, but I have listed them and included the citations of the papers that have shown these mechanisms. And here is a whole other set. And so the peer-reviewed data for the use of deuterium-depleted water in cancer patients are these. And so, the production and the shipping cost of the deuterium-depleted water make it a challenging intervention to maintain. And this is why the food, light, and other developing ways of using nature to reverse submolecular dysregulation are so critically important. And there's so much more to come. Stay tuned. So, in closing, I would like to end with this appropriate quote. One can know a good theory from a bad one by the formers leading to new vistas and exciting experiments, while the latter mostly gives birth only to new theories made in order to save their parents. This was spoken by Albert St. Georgi in the Introduction to Submolecular Biology in 1960. Thank you. Thank you for listening. I thank my patients for their excellent questions and their generosity. I thank Sophia Dorfsman for all her beautiful designs and figures. And I thank Dr. Laszlo G. Boros for sharing all of his wisdom, and I am especially grateful for his patience in explaining these mechanisms and findings. Without him, this presentation would not have been possible. So thank you.